Today we are going to discuss about how to handle cell lines. First, we will on the UV light. Remove all the aliquots and media bottles from the water bath. Clean it, wipe it with 70% alcohol. Open the sash at appropriate height. Wipe your hands with 70% alcohol. Keep all the media bottles, aliquots inside the hood after wiping with the 70% alcohol. Remove paraffin. Take one sterile falcon. For most of the saline, we will be using 10% DMA. Now we are taking 5 ml of FBS. Now we will add DMEM up to 50 ml. Close all the containers properly and tightly. Mix the media properly. Label it with the name and date of preparation. Wipe your working area with 70% alcohol. Now we will see the passaging of the cells. Remove confluent T25 flask from the incubator. Remove media from the T25 flask. Add little amount of PBS to remove the traces of FBS. It will hamper the activity of the trypsin. Mix it evenly. Remove the PBS. Now add 1 ml of trypsin to remove the cells. Detach the cells. Spread it evenly. Keep T25 in incubator. Bake for 5 minutes. You can observe under microscope to confirm the detachment of the cells. After 5 minutes, add 10% of DMA to inactivate the trypsin. Mix it properly. to detach the cells. We are inactivating the trypsin because trypsin will cause the cell death. Excess amount of trypsin will cause the cell death. Collect this cell suspension in sterile falcon. Centrifuge it at 1200 rpm for 5 minutes. This is a trypan gun we will be using for counting the cells. Take 10 microliter of trypan in one Eppendorf tube. This is the Nimbus chamber we are using for cell counting. Clean it properly. Place cover slip on. Remove cells falcon after 5 minutes. You can see the pellet. Remove the soap tank. Don't disturb pellet. Add 10% of DMEM. Displace the cell pellet in 10% of DMEM. Mix it properly to avoid the clumping of cells. Make it single cell suspension by pipetting for 20 to 30 times. Take 10 microliter of cell suspension for counting. Mix it properly, otherwise it will hamper the counting of the cells. Count the cells. At 10x. Add, add 10% media into the T25 flask. Add some amount of cell suspension into the T25. Close it properly and label your T25. Keep it inside the incubator. Take required amount of cell suspension as you have counted. Then add DMM media. Add 10% of DMM in sterile falcon. Add 10% media into the 6 well plate. Add calculated amount of cell suspension into the 6 well plate. Every time mix it properly and then add for even distribution of the cells. Close the 6 well plate and spread the cells evenly